Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today we are going to talk about transcription regulations. To discuss this topic, we have with us our subject expert, Dr. Charu Dogra Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
So today we'll be talking about transcription regulation. In fact, we'll be continuing with this. We were talking about transcription regulation in prokaryotes. The predominant level of gene regulation is at the transcription initiation, but the transcription regulation can also occur at transcription termination in prokaryotes. So the learning objectives for this session would be to understand the regulation at transcription termination in prokaryotes, and we will try to understand this by taking an example of a tryptophan operon, and then understand the cis regulatory components of transcription in eukaryotes. So we will also further our knowledge about transcription regulation by studying it in the eukaryotic system, and we will talk about the cis regulatory components of transcription regulation. So we start with the point that regulation can occur at the levels beyond transcription initiation also, and the two terms which we need to get accustomed with is the attenuation, which is a premature transcription termination that occurs in many amino acid biosynthetic operons, such as the one we will study today, which is the non-essential amino acid tryptophan synthesis, and the anti-termination that involves the formation of anti-terminator sequences or structures that prevents the formation of the terminator, resulting in transcriptional read-through into the structural genes of the operon. So the transcription stops when there is a termination sequence there and it is transcribed into RNA and it takes a form of a terminator structure. But anti-termination involves the formation of an anti-terminator structure so that the transcription continues and covers the transcription of the entire structural genes. So we look at the regulation at transcription termination by detailing the tryptophan operon. The structure of the tryptophan operon looks like this, where there are 5 genes which are involved: the tryptophan E, D, C, B and A. Tryptophan E and D encodes for the anthranilide synthetase, which catalyzes the first 2 steps of the synthesis of tryptophan from the corismic acid, which is shown in the lower panel of the diagram. The tryptophan C catalyzes the next 2 steps and it codes for the indole glycerol synthetase, and then tryptophan B and tryptophan A codes for the tryptophan synthetase, which forms the units that catalyzes the final step of the synthesis of tryptophan from the corismic acid. All these 5 structural genes are located or formed in the form of an operon with a regulatory component ahead of it. So if we look at this control region, then it contains a promoter and an operator, which usually overlaps with the promoter, but it also has this leader and an attenuator sequences lying in between the operator and the tryptophan E, the first structural gene of the operon. As you can see, this leader and the attenuator give rise to a mRNA. The attenuator is basically the part which will form a row independent transcription terminator, and then it codes for a 14 amino acid leader peptide. So we will look at how this change occurs or anti-termination occurs when the tryptophan is there. So basically the regulatory protein of the tryptophan operon is a tryptophan repressor, and it is an aporepressor, which basically means that by itself it is inactive. It needs the presence of a co-repressor which will activate this tryptophan repressor to do the job of the inhibition of the operon. The tryptophan repressor looks something like this. It is a tryptophan R dimer, which is shown as yellow and red, and it is in complex with some black molecules, which is a tryptophan molecule that act as the co-repressor, and you can see that it is bound to the tryptophan operator region of the DNA. So that is how the tryptophan repressor looks like. So the negative regulation of the tryptophan operon which occurs is basically that tryptophan R is the regulatory gene that codes for this protein tryptophan repressor. It is located far away from the tryptophan operon and not like lac operon, where the lac-I gene was somewhere upstream and near to the lack operon. But tryptophan R gene lies far away in the genome and it codes for tryptophan repressor. This tryptophan repressor, as I already told, is an aporepressor. By itself it is not active, but when there is a high concentration of tryptophan, which is a co-repressor, then the high concentration of tryptophan, the tryptophan molecules bind to this tryptophan repressor, and you can see that some conformational change has occurred in the blue repressor, and now it is able to bind to the operator sequence. Now this operator sequence is again overlapping with the promoter sequence, and therefore the RNA polymerase can bind but cannot effectively code for the entire gene. So it does bind, but it only give rise to a attenuated mRNA or the leader mRNA only. When the concentration of tryptophan is low, then the tryptophan is not available, and therefore the repressor remains in its native state, which is the repressed, which is basically inactive state, and therefore it is not able to bind to the operator, and therefore it is not able to shut down the synthesis of the tryptophan. So low concentration of tryptophan ensures that the tryptophan mRNA is transcribed, and therefore the E, D, C, B and A proteins are formed so that the synthesis can occur, but the high concentration of tryptophan ensures the shutdown of the synthesis operon because it does not need any tryptophan, the concentration is already high. So this is a typical negative regulation of the of any operon or negative regulation mechanism which occurs in the tryptophan which is there. But over and above this, there is another mechanism which occurs, which is called as attenuation. And this attenuation or premature transcription is based upon the structure of this leader mRNA. If you look at this leader mRNA, which is the tryptophan L, then you can see that now the positions are shown where there is AUG, the start codon, and then there are two tryptophan codons which are there. There is a UGA, which is a stop codon. Then there is a place which is shown as TXN pause, and we will look at that the transcription will pause at this point, and then there is an attenuator region which is there. When it is encoded into the mRNA, and we see that it contains 4 regions which are showing complementary to each other. So 1 is complementary to 2 and 3 is complementary to 4, but 2 and 3 are complementary to each other also. So when there is a higher concentration, so basically these can attain 2 different kind of structures as it is shown in the diagram. It can attain the structure where 1 and 2 have formed a stem loop structure and 3-4 region have formed a stem loop structure, and such a thing is called as terminator, because this 3-4 structure will act as a terminator and will lead to the termination of the transcription. But there is also a possibility that 2 and 3 complementary with each other forms a stem loop structure as shown in the right hand side of the diagram, and that is basically the terminator structure is not formed. So 3-4 is not formed, and therefore that is kind of an anti-terminator structure, and if an RNA polymerase encounters such a kind of a structure, it will not terminate the transcription and it will continue, and therefore this is anti-termination will occur. So this is how the tryptophan leader mRNA looks like, and that is how they can alternatively find different complementary sequences, and therefore they can form different kinds of either terminator or an anti-terminator structure. Now the formation of the terminator and anti-terminator structure is governed again by the presence of the tryptophan which is there. So transcriptional attenuation of tryptophan operon, for example, if there is low level of tryptophan, if it is there. So the low level of tryptophan will ensure that when the basic mechanism is that in its point to understand is that in prokaryotes the transcription and translation is happening simultaneously. So co-transcriptional translation is occurring as the RNA is coming out, the ribosome is sitting on it and translating it into polypeptide also. So when the mRNA is there, and from this mRNA, when the ribosomes bind to it and starts translating it, then it encounters or reaches the point where the 2 tryptophan codons are there. So we just saw that in the leader peptide, these 2 codons are residing in the region number 1. So if there is low level of tryptophan, then if you review or you will be studying about the translation process, that the tRNA gets the amino acid which is to be incorporated into the protein, into the growing polypeptide chain. Now tRNA loaded with the amino acid, with the cognitive amino acid, is said to be charged. So, charging of the tRNA depends upon the availability of the amino acid which is there. So, if there is low level of tryptophan, there will be low concentration of the charged tRNAs with the tryptophan amino acid, and there will be more of uncharged tRNAs. So, when the ribosome is moving along translating the mRNA, the leader mRNA which has come out, then at tryptophan codons it will stall. It will wait for a while because the tryptophan, the charged tRNA are less. So, it will stall at that point, and by that time the region 2 and 3 of the mRNA, because they are complementary to each other and RNA polymerase has already transcribed that region, will form a stem loop kind of a structure. Now, you know that this 2-3 complementarity stem loop structure is basically an anti-termination region. So, the ribosomes as well will not stop here and they will continue, and the RNA polymerase will also continue, and this will act as an anti-termination region, and the entire synthesis of the entire mRNA, polycistronic mRNA will occur. The proteins coding for the genes will form, and therefore the tryptophan synthesis will continue. So, low level of tryptophan will make sure that the premature transcription does not occur and the entire transcription of the entire tryptophan operon happens, and therefore the tryptophan synthesis can occur. But if you look at the another condition when there is a high level of tryptophan which is there. So, when there is a high level of tryptophan, then the ribosome would not stall actually at the region number 1, because it has plenty of charged tRNA which is present inside the cell. So, the ribosome will quickly move away from the region number 1 and will also encompass the region number 2 which is there, because it is quickly moving. So, when it encompasses the region number 1, it contains the region number 1 and region number 2, then region number 3 and 4 are left because it has been transcribed by RNA polymerase, but they will attain a typical terminator structure. They will attain a typical stem loop structure which will act to terminate the transcription. So, if there is a high level of tryptophan, then the terminator structure will form and the RNA polymerase will only be able to synthesize the leader mRNA peptide, and the entire tryptophan polycistronic mRNA is not transcribed. So, the level of tryptophan, apart from regulating by negative regulation by the action of tryptophan repressor, has an additional system of regulation by this premature transcription that occurs, the premature transcription termination that occurs when there is a high level of tryptophan. However, when there is a low level of tryptophan, then the transcription of all the genes occur and therefore all the polypeptides are formed and the synthesis of tryptophan takes place. So, this regulation is only present in prokaryotes at the transcription termination level. So, now we move on to the next point, which is the transcription regulation in eukaryotes. So, we have now detailed the transcription in prokaryotes, but now we will talk about the transcription regulation in eukaryotic system. And to start with, we will just compare the gene regulation strategies in prokaryotes and eukaryotes. The difference is that the eukaryote contains a nucleus. So, the genes are transcribed inside the nucleus, but from the nucleus the mRNA has to now come out into the cytoplasm and translation occurs in the cytoplasm. In the prokaryotic system, the transcription and the termination and the translation was occurring in the same compartment, but in eukaryotes this is compartmentalized. The transcription occurs inside the nucleus, however translation happens outside the nucleus in the cytoplasm. Therefore, an additional level of regulation is imposed here, which is for the export of mRNA from the nucleus to the outside. Then there is a coordinated cis regulation that occurs. Now, there is an absence of coordinated cis transcription in the eukaryotes. And what does that mean? That because there are no generally there is an absence of operon system, because the translation cannot start from, generally there are exceptions, but generally the translation cannot start from the internal mRNAs. Therefore, there is not generally not a polycistronic mRNA which is formed, and therefore the translation cannot occur in a coordinated manner for all the genes like it used to happen in the prokaryotic system. So, there is an absence of coordinated cis transcription. However, the coordinated cis regulation is still possible due to the sharing of the distant regulatory elements which control a number of genes which are lying. They can control a number of genes, the common regulatory elements, as we will see. Then the presence of introns also allows the regulation at a step between the transcription and the protein synthesis, which was not there in prokaryotes, because the selective information of the gene can be used, and therefore there is a mechanism which is called as alternative splicing, which will lead to the formation of different mRNAs. So different mRNAs can be formed and the intervening sequences can be removed and exons are usually joined. So this allows an additional step of regulation in case of eukaryotic system. Then the regulation of gene activity by chromatin structure is present both in prokaryotes and eukaryotes, but in eukaryotes it is tremendous, and this allows large sections of DNA to be repressed or activated en masse. So basically we will see that because the the higher order of genome organization in case of eukaryotic system, there are nucleosomes which are present. So the regulation of gene activity also involves chromatin modulations, and in fact this can be on a wider scale, and entire area of a chromatin can be repressed, as we will see in the case of heterochromatinization, or the active regions can be totally in a group, they can be activated also. So chromatin regulation plays a lot of an important role in case of eukaryotes. Then the RNA polymerase activity. The efficient transcription requires constitutive positive regulators to stabilize the initiation complex. In bacteria, we saw that the RNA polymerase, the sigma subunit used to give the specificity for the promoter and the transcription used to start. But we have seen in eukaryotes that it is a very elaborate mechanism. The RNA polymerase by itself cannot bind to the promoters. There has to be a plethora of general transcription factors sitting on the promoter, and then the RNA polymerase is recruited, and this is not a very stable thing which happens. So constitutive positive regulators are needed for the stable initiation to be there and the transcription to be very efficient. So that is another additional point in case of eukaryotes which is needed. Then if we look at the transcription regulation, then the regulation of transcription often depends upon the factors which bind at a distance from the basal promoter, and interaction is mediated by looping out of the intervening DNA. So we will see that how this basically occurs. Then post-translational modification of TF is also a very common regulation strategy in case of eukaryotes. The de novo synthesis of new transcription factors is used for relatively slow and lasting responses. So we will see the regulation of transcription factors itself, that it can be regulated at a post-translational modification. That means the transcription factors are constitutively present, which is a little less efficient because the resources are wasted for the transcription of the formation of the transcription factors even when they are not in use. So the post-translational modification, but it is needed for the quick response, because if for something you need quick action of the transcription factor, then to start from the transcription initiation till the formation of the transcription factor in a particular concentration so that they can effectively upregulate or downregulate the transcription, it takes time. So for quick responses, post-translational modification is regulation is important, but for late and for differentiation and usually in the developmental processes, the de novo synthesis, when it is needed, the transcription factors are synthesized. Then that happens in case of eukaryotic systems, and we will elaborate on this further down the through the lecture. So we talked about the class 1 genes and class 2 and class 3 genes which were transcribed by different RNA polymerases in case of eukaryotes. The class 1 genes which code for the 5.8S, 18S and 28S ribosomal RNA genes are constitutive in nature, and the class 2 genes are all protein coding genes and most genes for small nuclear RNAs, as well as class 3 genes which coded for tRNA genes, 5S rRNA genes and gene encoding the small nuclear RNA and various small cytoplasmic RNAs, they are all regulated. Now we will look at the regulatory components of the transcription. There are cis-acting elements and there are trans-acting factors. So the regulatory cis-acting elements are basically the promoter proximal elements, the enhancers, the silencers, the locus control regions, and the insulators or the boundary elements. So, we will talk in detail about all these elements. They are cis-acting elements because they localize at the same locus of in the promoter present on the same chromosomes, and they are regulatory in nature. So, promoter proximal elements are the sites located 5' to the basal promoter in the upstream promoter region, usually between positions between -50 to -500 base pairs. So, you can see there is a core promoter. For example, it has a Tata binding element which is there. So, there is a core promoter, and promoter proximal elements lie somewhere upstream in the upstream promoter region, approximately from -50 and -500 bases. It is required for high levels of transcription. So, the core promoter can give a basal or minimal level of transcription. The binding of regulatory molecules at promoter proximal elements will give rise to a higher, or actually the true level of transcription which is occurring in the cell. So, for the basal or minimal level of regulation, only the core promoter is required, but promoter proximal elements and the binding of the regulatory proteins on these elements are necessary for a true level of transcription which is shown by the genes. They generally function in either orientation, and the examples include, for example, which is called as the CAT box, the CAAT box consensus sequence, the GC consensus sequence is given, the GC box, and some octamer consensus sequences are there. So, these CAT box, the GC rich box, they are all promoter proximal elements which are present. Various combinations of core and proximal elements are found near different genes, and basically, as I said, that they are very keen for, they are the key elements for the gene expression because they are required for the gene to have a true level of expression. The activators, the proteins important in transcription regulation, are recognized by these promoter proximal elements. For the housekeeping genes, which are constitutive in nature, they are used in all cell types for basic cellular function. For example, the genes encoding the enzymes for glycolysis. They have common promoter proximal elements found across all the cells, and they are recognized by activator proteins found in all the cells. The genes expressed only in some cell types or at a particular times have usually promoter proximal elements recognized by activator proteins found only in the specific cell types or times. So, we are saying activators all the time. Basically, it means the regulatory proteins but the predominant regulation, the constitutive regulation which is required is activation. So, we are talking about activators here. So, the promoter proximal elements will give rise to the true level of expression. So, for the housekeeping genes, they will be same in all the different cells, but in different cells, when the different specific cell types, specific cells have to be transcribed, then they will have cell type specific promoter proximal elements which are there. Then there are enhancers which are required for maximal transcription of genes, and somewhere like 1000-fold increase in the level of transcription, and they can be located upstream or downstream of the transcription initiation site. So, they are position independent, and they modulate from a distance of approximately greater than 50 kbps. So, they are very far off away from the initiation site, and because they are very far off, the interactions is usually mediated by looping out of the intervening DNA, allowing them to act in a position and orientation independent manner. So, they are lying very far off on the away from the promoters, they are going to regulate, but looping of the DNA will bring them nearer to the transcription, the promoter transcription initiation machinery, and then they will regulate. Usually they do not directly, the activators bound to the enhancers do not directly affect the basal initiation apparatus or the RNA polymerase, but requires the presence of a mediator, which we already talked about earlier. The distant regulatory cis elements that act negatively to repress the function of a gene are called as silencers. So, enhancers upregulate the regulation, and the silencers negates the regulation or diminishes the regulation, but they are usually the distant acting cis acting elements which are regulating the transcription. Then there are some things which are called as upstream activator or upstream repressor sites. They are enhancer or silencer like sites which are found in yeast that function in a similar manner, but they are incapable of operating downstream of the gene they control. So, they are usually present, they are always present on the upstream of the side of the promoter, but they can be, they are distant. So, they are like promoter proximal, they are elements in the sense that they are lying upstream to the promoters they regulate, but they can lie, they are similar to enhancer or silencer like, because they can act from the many base pairs away from on the DNA, located on the DNA. Another kind of cis element is called as the locus control region. They are distant regulatory elements controlling the gene expression by establishing an open chromatin domain. In fact, they were identified by the fact that they work in the manner to enhance the transcription. So, what they did was, when they transfected it into the cell line, and when the gene, for example, the beta globin gene were transfected into the erythroid cell line, then it enhanced the transcription if the promoter and enhancer was there. But if this construct was introduced into a mouse genome, then it was not able to enhance the transcription, not even in the erythroid specific cells. And then they found out that then if this locus control region was included with the construct, then it was basically upregulating the gene expression in the erythroid cell. So, without this locus control region, so it is not just acting as an enhancer, because if it is not introduced if they are introduced into the transfected cells, then they are not able to enhance the transcription, because probably they act by showing their effect on the organization of the chromatin, rather than acting just like an enhancer by binding of the protein and modulating the activity of the transcription machinery. So, they alleviate this cis repression caused by the chromatin silencing, allowing position independent and copy number dependent, that they also found out that depending upon the copy number of the construct, the transgene that was introduced into the cell line, the effect was based upon that, but they do not enhance the transcription activity in a transient transfection assays in the manner of a classic enhancer. So, they are not just enhancers, they are not acting by modulating the activity of the transcription machinery or by increasing the upregulating the way the enhancer acts, but they act at the level of the chromatin organization, the higher order chromatin organization, by modulating or by enhancing the formation of the open chromatin domain which is there. So, locus control regions are basically found upstream to the cluster of globin genes. These are the beta globin genes which are there, and LCR can be found. Then the mouse beta globin locus is also shown. Then these are the LCR. So, the genes do have, like for example, beta globin gene has dual enhancers, but the locus control region is the region that enhances that alleviates the cis repression which is present. So, if you look at the cell nucleus, now we talk about the chromatin domains, and that chromatin domains is determined by the transcriptional activity of the genome region. So, you can see that there is an active portion and inactive portion, and usually what we say that the functionally distinct regions are separated. So, now we have done the chromatin genome organization, and we have seen that the chromatin is thrown into loops, and then the loops are scaffolded on the nuclear matrix. So, the regions where they are scaffolded on the nuclear matrix probably represent certain sequences which are separating different loops. If we consider these loops as domains, the functional domains, then these sequences are actually differentiating between different domains, and these sequences are termed as insulators or boundary elements. So, I show this global view of the cell nucleus just to make you understand the idea of the chromatin domains, and then these chromatin domains are of additional things are superimposed on these super domains, and then they again give rise to the chromosomal territories which are there. So, that is how the chromosomes are organized inside a nucleus. So, insulators or boundary elements are the sequences that map near the border of biochemically defined chromatin domains. So, they are functionally distinct, and probably one domain cannot alter the function of the second domain, and these sequences which probably are the if we consider in terms of structure, then they are the sequences which are scaffolding the loops onto the matrix. So, they act as the insulators or boundary elements, and they should be able to isolate the genes from the effects of the adjacent domains as shown in the diagram. So, an enhancer which is bound by the activators can influence the activity of a promoter, but if an insulator is present in between, then this enhancer cannot modulate the activity of the promoter. Then in the third line you can see that the enhancer is influencing the activity of a promoter which is lying on its left hand side, but still it cannot act on the right hand side promoter because there is an insulator or a boundary sequence element is there. And again in the last it is shown that a different enhancer is needed, that is modulating the activity of the promoter which was on the right hand side, but the left hand side cannot see. So insulator or boundary elements are basically differentiating the chromatin into different functional domains, where one domain cannot influence the function of or cannot regulate the function of the genes which are present on the other domains, and therefore far reaching enhancers cannot just act on any promoter which is lying in its vicinity. The insulator mediated loops may segregate individual loci containing the coding part of the gene and its regulatory regions from the surrounding genome landscape with other regulatory elements. So these insulator mediated loops are formed. So there are two insulators which are shown, and in between the transcription unit which has an enhancer and a promoter. So they will form a loop by the binding of the insulator proteins, and the transcription factor and RNA polymerase will modulate the activity of the promoter which is lying between these two insulators, but they cannot work on the transcription of other genes which might be present on the other things. So insulator mediated loops are also the structures which can be formed in this particular manner. Thank you. So we continue, we start with the transcription regulation in eukaryotes, and we will study in this particular session about understanding the transregulatory components of the transcription in eukaryotes, and we will look at the mechanism of action of these transregulatory factors. So cis-acting elements we have talked about, in which promoter proximal elements, enhancers, silencers, and insulators and boundary elements or locus control regions, how they were responsible for the regulation part. And now we will look at the transregulatory components or the transacting factors. So the transacting factors can be transcriptional activators that will upregulate the transcription, and transcriptional repressors that will downregulate the genes, or they will repress any kind of transcription which is there. Now transcription factors usually possess several domains. The two important domains are the DNA binding domain, by which these transcription factors bind to their DNA, they bind to the elements on the DNA. They also contain an activation or a repressor domain, which will the activity center of these particular proteins, but they often also contains dimerization domain, as we have seen that transcription factors usually work by the formation of dimers, whether homo or heterodimers, and the domains for interaction with effector molecules or signal transduction components. For example, with they interact with the mediator molecules, or they have to interact with other effector molecules which will modulate their activity for the upregulation or downregulation of the promoters they regulate. So, the range of DNA binding domains, we will just have a look at the different type of nucleic acid or DNA binding domains which these transcription factors possess. The proteins often bind as dimers and recognize specific DNA sequences using an alpha helix inserted into the major groove. This is quite similar to what we studied in case of prokaryotes, when the lac repressor or the catabolite activator protein used to have a helix turn helix motif, in which one of the helix was termed as recognition helix, that was introduced into the major groove of the DNA, and such a kind of a DNA binding domain was present in them. The eukaryotic transcription factors also contain such kind of motifs. We will have a look at them, but they bind as heterodimers, and that these heterodimerization extend the range of DNA binding specificities which is available. So, one of the binding domains is termed as the homeodomain proteins, and you can see that homeodomain proteins is again belongs to the class of helix turn helix, but it consists of 3 alpha helices as shown in the figure by numbered as 1, 2 and 3. The 2 and 3 form the structure resembling that helix turn helix motif. However, helix 3 is the recognition helix and it is inserted into the major groove. You can see that this helix 3 makes base specific amino acid specific, base specific recognition in the major groove element. In fact, an arm of the helix number 1, alpha helix 1, also extends a tail that can make interaction with the bases, which is there for further stability. The homeodomain proteins were discovered in Drosophila, but they are also found in yeast, which is there. Then there are certain domains which are such as zinc containing DNA binding domains. They incorporate zinc atoms, and the examples include the zinc finger protein, for example the transcription factor 3A, which was required for the transcription of the class 3 genes, and zinc cluster domain. The zinc is coordinated by 2 histine in the alpha helix and 2 cysteine in the beta sheet. So, you can see that there is a alpha helix which is there and there is a beta sheet which is there, and the zinc atom in the center is coordinating with 2 histine residues on the alpha helix and 2 cysteine residues which are present in the beta helical region, and sometimes it is by 4 cysteine residues just to stabilize the structure, and this alpha helix is basically the recognition helix. So, such kind of a zinc containing DNA binding domain also the transcription factors also utilize such DNA binding domains for interacting with the DNA. Then the next is the leucine zipper motif, and as you can see, the 2 large alpha helices form both dimerization and DNA binding domain. So, they have a pincer like structure. So, the 2 alpha helices dimerize also, and they also bind to the DNA. Each alpha helix inserts into the major groove. Towards the top, the 2 helices interact to form a coiled coil, wherein 2 helices are held together by hydrophobic interactions between the appropriately spaced leucine residues. So, usually they can form hetero and homodimers. So, you can see that the 2 alpha helices on the top, because they are appropriately placed leucine residues are interacting. So, they appear like a zipper, and therefore, it is said to be a leucine zipper motif. The other motif which is present, which is common, is called as the helix loop helix proteins. In this extended alpha helical region, from each of the 2 monomers is inserted into the major groove. So, as it is shown in the picture, there are 2 alpha helices which are there and they are inserted on the 2 sides in the major grooves. The dimerization surface is formed from the 2 helical regions. One part is involved in the DNA recognition and the other is usually a shorter alpha helices. The 2 helices are separated by a flexible loop. This is often called as the basic zipper or the basic helix loop helix protein, because the region that binds the DNA contains basic amino acid residue. So, usually it is represented as BHLH residue, where the DNA binding region of the alpha helix contains more amount of basic amino acids which is there. So these are the DNA binding motifs which are there. So there are a particular number of particular types of DNA binding domains which can exist in different transcription factors. Activating region, if we look at, however, are not very well defined structures. They are grouped not on the basis of the sequence, or because, as I said, they are not well defined structures, so they are not structurally differentiated, but they are grouped on the basis of the amino acid content which they have. The acidic activation, so they are divided into three activating families, domains. One is the acidic activation domains. For example, the yeast GAL4 transcription factor belongs to this category, and mammalian glucocorticoid receptor. They contain up to 20% of glutamic and aspartic acid residues, and they can activate transcription when bound at the promoter or a distant enhancer. So these acidic activation domains are very common in the activator proteins which bind to either the promoter elements as well as from the distant enhancer elements they bind. The other kind of activating region based upon the activating domain is the glutamin rich activation domain. For example, it is found in the constitutive transcriptional activator SP1. They are also, but they appear to be unable or only weakly able to activate transcription when bound to distant enhancer. So in fact, the next two domain families, they are weakly able to enhance the replication when bound to distant enhancers. So they work in proximity, they are found in the transcription factors which are which bind near to the genes, the promoters they activate, they regulate. So there are proline rich activation domains which are there. They are found in the proteins such as c-Jun and the CCA-AT binding factor CTF and NF1. They appear to be unable again or only weakly able to activate transcription when bound to distant enhancer. So activating regions can be can be grouped into these three different kind of domains based upon the amino acid content. But these are the activators we are talking about. The defined inhibition domains are usually proline rich and they generally lack charged residues. However, few direct transcriptional repressors are found. We will see when we talk about the mechanism of regulation that the transcription repressors usually work by stopping the activator to do its job. So they are not directly repressing by binding to the DNA sites or modulating the activity of the RNA polymerase or the transcription machinery, but they generally work by inhibiting the activators to activate the transcription. So there are very few inhibitory direct transcriptional repressors which are there, and therefore they do not need to activate or bind to the DNA. So the defined inhibition domains are usually proline rich and they lack the charged residues which are there. So this is all about the transcription factors, the transregulatory factors, how they bind to the domain and how they activate or repress the transcription mechanism. Now we will go into a little detail about the mechanism of action of these transcription factors. In principle, the transcription factors and that acts in basically four ways. They can do so by binding to the DNA at particular sites and then interacting with the basal initiation apparatus. So we already know that in eukaryotes there is a pre-initiation complex of certain constitutive GTFs which are there, which is formed at the core promoter, and then the RNA polymerase is also recruited and that pre-initiation complex then interacts, so activators usually interacts with this PIC or the regulatory components interact with this basal apparatus to modulate, to regulate the downstream promoter, and they do bind to the DNA. Another way is that they, by binding to the DNA and altering its structure or conformation, so instead of acting on the proteins which are bound to the DNA, they change or they modulate the conformation of the DNA itself. So the regulatory proteins can bind to the sites on the DNA and alter its organization and therefore regulate the transcription of the genes. So that is also one of the mechanism. Then transcription factors can also act indirectly, that is by binding to the DNA and instead of interacting with the initiation apparatus or the DNA, they interact with another regulator to influence its activity. So they do bind to the DNA, but they modulate the activity of the other regulatory molecules which can affect the transcription of the genes. Then the last way is that they can also work by protein-protein interactions, that is without binding to the DNA at all, that they only either directly with the components of the basal apparatus or indirectly with other regulators. So by protein-protein interactions, the regulatory proteins interact with the proteins of the basal apparatus or other regulatory molecules and regulate the gene expression. So in principally, these are the 4 ways by which this transcription factors can work. So we will look at that how the transcription in eukaryotes is modulated by the interaction with the pre-initiation complex which is there. So activation domains of all the 3 clauses stimulate the recruitment of the TF2B to the initiation complex. So we are talking about here the transcription of the class 2 genes and transcription factor 2B, which is sequentially bind second after the transcription factor 2D is bound to the Tata binding protein, then there is a BRE element where this TF2B binds. So activation domains of all the 3 classes stimulate the recruitment of this TF2B. The acidic activation domains induce a conformational change in TF2B which increases its affinity for the RNA polymerases. So this is how the transcription factors are interacting with the proteins of the pre-initiation complex and modulating. So we are generally talking about the activation here. The Tata binding protein is also a target and activation is mediated through the interaction with the Tata binding associated factors or the TAFs. Different TAFs interact with different TAFs and different TAFs assemble on promoters with different architectures. So how they regulate? That different transcription factors can associate with different transcription associated factors, and in fact this TF2D is formed from Tata binding protein and different set of transcription associated factors which can be there, and therefore they can assemble on the promoters with different architecture and therefore transcribe different kind of genes which are there. A multi-component complex, we already talked about, the mediator assembles on the RNA polymerase 2 C-terminal domain to transmit the signals from the transcriptional activators. So that is how basically the activators sends the information to the promoters by the RNA polymerase by this multi-component complex called as the mediator. Then some direct repressors, as we talked about, have been isolated, although they are few in number. They interact directly with the basal initiation complex. For example, there is a protein called as DR1 that inhibits assembly of the pre-initiation complex. Other repressors have discrete cis-acting sites in the promoter or in distant silencer elements and may act either to inhibit the assembly or the activity of PIC. For example, the Drosophila pair rule protein called as even skipped and the mammalian thyroid hormone receptor, in the absence of its ligand, work by this mechanism. They have discrete sites near the promoter or at a distant silencer elements, and then they inhibit the assembly or activity of the pre-initiation complex. So transcription factors may interact with this pre-initiation complex to either activate the transcription regulation, and some direct repressors can again prevent its assembly and therefore can downregulate the expression of the gene. The next mechanism which we look at is that the transcription factor can also work by altering the DNA structure. They can displace the nucleosome. They can do so and therefore directly activate by permitting the binding of other transcription factors in the near vicinity, which has been previously blocked by the nucleosome positioning. So they might, unravel certain binding sites where the transcription factors can now bind and enhance the transcription of the genes. Or they may also facilitate rearrangement of the chromatin in the promoter and alleviate repression caused by the higher order chromatin structure. So we will see that a gene silencing can be occurring by the organization of the chromatin. So if it gets heterochromatinized, the gene activity is repressed. So the nucleosome, or the displacing the nucleosome, or nucleosome remodeling as it is called as, so this nucleosome alterations can actually alleviate the repression which has been imposed due to the organization of the higher order structure in the DNA. So they can take away that and probably enhance the replication. So there are two ways of nucleosome remodeling. First is the chemical modification by enzymes such as histone acetyltransferase. They alters the interaction between the histone tail and the nucleosome, and transcriptional machinery will have higher affinity to the promoter. So certain enzymes such as histone acetyltransferase can modulate the histones, can cause them to chemically modify and therefore enhance or therefore increase the transcriptional machinery to have high affinity for such promoters. Another method of nucleosome remodeling is that the remodeler works on them, then they increase the mobility of the nucleosomes, freeing up the binding sites for the regulatory and the machinery. So the basic idea is that the transcription factors alter the DNA structure to loosen the chromatin, or to basically create or to basically expose those regulatory sites where the transcription factors can bind and alter and regulate the gene expression. So that is also one of the mechanism. The examples are the GAGA factor in Drosophila and the SW1 SNF complex associated with the C-terminal domain of RNA polymerase II in yeast and vertebrates. They work by altering the DNA structure. Some TFs bound at enhancers and silencers can act similarly by propagating a chain of chromatin organization over a wider area. For example, the yeast mating type cassettes HML alpha and HMR alpha are kept in an inactive state by heterochromatinization directed by silencers located several kilobase pairs away. So not only the promoter proximal, the activators that can bind to the promoter proximal elements can exert their function by altering the DNA structure, but also the regulatory proteins which are bound to enhancers or silencers which are many kbps away from the promoter can influence in the same manner. Then some transcription factors primarily work by introducing bends into the DNA. Now HMG box transcription factors such as SRY, which controls sex determination in mammals, SOX2, SOX3 and SOX11 which are involved in neural differentiation, and LEF1 which controls lymphocyte specific gene expression. They can organize the DNA into a specific 3D conformation wherein all the various transcriptional activators interact productively with each other. Now the transcriptional activators which are bound as far off, we have just seen that DNA looping and bending is very important. So the transcription factors binding sites might be present in between the enhancers and the promoters where these transcription factors can bind and actually introduce the bends. So the bend will bring the activators onto near to the promoters they regulate, and therefore they can regulate such gene expression in there. So that's about the altering the DNA structure. Then the transcription factors can also interact with other transcription factors to regulate. Most eukaryotic transcriptional repressors, as I have already said, act passively by interfering with the function of the activators. So the transcription repressors work by affecting the other transcription factors which are the transcriptional activators. Now one mechanism to do so is to simply occupy the binding site of the activator. They either overlapping or adjacent, they bind, and therefore the steric hindrance prevents the binding of the activator. So they bind either onto the activator binding site or the site which is overlapping to it or just next to it, and therefore the activator site bound by the repressor the activator will not be able to bind to it then. So this reflects that there is a kind of a competition between the activator and the repressor for the binding site. The example of regulation by such competition for binding sites is provided by Drosophila even skipped gene, which is regulated by competition between the various gap proteins for occupation of the upstream binding site. So we see it in the case of Drosophila that such a kind of mechanism occurs. Then the indirect silencing, which is termed as quenching, can also be there, where an inhibitor binds to a site in DNA which obstructs the activity of a bound activator, often by blocking its access to the basal apparatus or masking the activation domain. So we have seen how the activators, when bound to their binding sites, can modulate the promoters. So they can interact with the pre-initiation complex, or they have certain activation domain which will bound to the other regulatory components and therefore activate the gene transcription. But the indirect silencing can be there when the inhibitors can actually bind to their activator activating sites, and therefore they obstructs the bound activator. The activator is able to bind to its activation site but still not able to exert its function which is there. The next aspect is that how these transcription factors are then regulated. The transcription factor can be regulated in two ways, by controlling their availability and controlling their activity. So, it's basically the thing is that the transcription factors are also proteins, they are also transcribed by genes. Now, for the transcription of the transcription factors, you need more transcription factors. So, there is a kind of a chain which is there. So, this chain can be modulated by two mechanisms, that either the availability of the transcription factors can be enhanced, or they are constitutively produced but their activity is controlled. And as I said, the two mechanisms are there. The second mechanism is predominantly in case of bacteria, although it is not efficient because the transcription factors are produced even when they are not used. So, the resources are wasted. But for quick responses, as I said, this method, the controlling by activity or post-translational modification is required, because it takes time for the information to, when the module regulation is at the initiation step, or by controlling their availability, their synthesis, there is a time by which if you have to increase the level of transcription, the information will go there and the sufficient amount of transcription factor is produced. So, there is a time lag which is basically there. The regulated de novo synthesis of transcription factors is useful when a long-term transition of transcriptional activity is required, and therefore, this method is prevalent in regulation of the developmental processes. The regulation of the transcription factor activity, as I said, allows a prompt response to the external stimuli. In some cases, the stimulus itself is in the form of a transcription factor. For example, in lactoferrin, it is released by neutrophils in response to bacterial infection. They can act as a transcriptional activator to induce a transcription of a number of genes by itself. In some cases, the stimulus acts like an allosteric modulator of the transcription factor, as we saw in case of bacteria, the lac repressor, or the allolactose, which is the stimulus, is basically acting as the inducer of the lac operon itself. In case of eukaryotes in mammals, the steroid hormones act as an allosteric modulators of their receptors, which are also the transcription factors. In some cases, the stimulus acts as a signal which activates a transcription factor. For example, the JAK-STAT pathway. The cytokine is a signal which binds to a receptor and activates the intracellular JAK kinase, which activates a transcription factor of the STAT family. So, there is an entire signal transduction pathway which occurs, and that regulates the transcription of the gene. In fact, this signal transduction also tells us another mechanism of regulation of gene expression in eukaryotes, which is the covalent modification. The phosphorylation and kinase. So, kinasing the enzyme kinase, they phosphorylates the downstream factors, and therefore information is passed on by this covalent modification, and finally the transcription factors inside the nucleus then bind to the downstream genes to either upregulate or downregulate the genes which are there. The last aspect in this is about the combinatorial and context-dependent regulation which is occurring in case of eukaryotes. So, combinatorial control is basically when the transcription factors work together in synergism to act for the activity of a gene. So, transcription activity of a gene driven by two transcription factors is greater than the additive effects of the individual factor. The transcription factor PIT1 acts synergistically with estrogen receptor in the prolactin gene expression. So, that is usually a combinatorial regulation that occurs. Then the context dependent regulation basically means that the two transcription factors which activate a gene independently cause silencing of the gene in combination. This regulation may be dependent on the abundance of a transcription factor or presence of specific regulatory sites in the DNA. For example, P53 is a strong activator of gene expression if there is a P53 recognition site in the promoter. But if there is no site, it binds to TF2D and inhibits the transcription. So, it can act as an activation, it can act as a regulation, but it is a context dependent regulation that is occurring. So, this is all about the transcription regulation, but the epigenetic part of the regulation and gene silencing or the strategies of transcription regulation we will take in another lecture which will be there.
On that note, I would like to thank ma'am for this very enriching lecture, and I would like to thank you, dear friends, for watching our show, and I hope that you will continue to watch our show in coming days as well. On that note, I would like to thank you guys, and stay tuned and keep watching. Thank you.